There's big news on the equine influenza front, and scientists with the Horse Genome Project are searching for the genetic origins of everything from coat colors to laminitis. I'm Erin Ryder, news editor of The Horse, Your Guide to Equine Healthcare. Welcome to the Ryder Report for November 21st. The independent inquiry into the history and management of Australia's equine influenza outbreak is uncovering flaws in the government's quarantine procedures, according to recently published transcripts. Since last Tuesday, inquiry leaders have interrogated the managers of the government-run Australia Quarantine Inspection Service and Eastern Creek Quarantine Centre, where the first infected horse was found. The managers have admitted that they ignored warnings, put off meetings, missing work instructions, and lack of managerial responsibility are some of the problems that led to the outbreak. The virus infected 40,000 horses in the first 9 weeks and caused a complete lockdown on equine movement in New South Wales and Queensland. The outbreak is estimated to have cost $3.94 million per day nationally. TheHorse.com is following this story. In the story that never ends, there's yet another outbreak of equine influenza. You might remember that we mentioned an outbreak of equine influenza in China during the November 14th Ryder Report. Well, this week Mongolian officials reported 7 outbreaks in that country. Interestingly enough, the outbreaks have occurred along the Chinese border. 9 horses are reported to be dead, and 11,600 are infected. Researchers in equine genetics received a gift last February when the completed DNA sequence of the horse was unveiled. Since it became available, the researchers with the Horse Genome Project, which is a cooperative effort by more than 100 scientists in 20 countries, have made great strides in researching coat color genetics and hereditary diseases that are caused by single gene defects. But they're also looking at more complex problems such as laminitis, polysaccharide storage myopathy, and recurrent airway obstruction.
For studies, this has made some approaches available that I didn't think would be available until after I was retired. And now we're faced with being able to do these experiments. And so people have been collecting families. The Morris Animal Foundation has awarded us a consortium grant for developing tools that we can use to study genetic variation and also looking at gene expression. And that will become available now. So a lot of it has been stage setting. We have a new website. We have a list of the diseases and the conditions that scientists are interested in looking at, and it's very broad.
One recently completed project on Tobiano coloring demonstrates some real-world applications for the research. We go now to the University of Kentucky's Gluck Equine Research Center, where Dr. Samantha Brooks will tell us more about her research on the coat pattern and a new test that will help breeders. I'm here with Samantha Brooks, a researcher at the Gluck Equine Research Institute. She's recently completed a project on the Tobiano gene in horses. Dr. Brooks, can you tell us a little bit about your project?
Sure, absolutely. Tobiano gene is a very popular spotting pattern. It's present in a wide variety of horses, primarily the paint horses, which we all think of, but also pintos, draft horses, ponies, and it's very popular because it's an aesthetic appeal. It makes them very flashy animals. So breeders really would like to increase the number of spotted foals that they produce, and in order to do that, they need to be able to look at the genes, because from the outside of the horse, you can't necessarily tell if a horse is going to be true breeding or not. So for years, we've had several different tests that we could use, different types of markers and things in order to look for the Tobiano gene, but they were all just markers. And for some families, these markers didn't work, and we occasionally get false positives and false negatives, and that creates an issue sometimes for the breeder who's investing in these tests and would like to have a product that they know is going to work. So for that reason, and as well as for our interest as scientists and as biologists, we decided to investigate that a little further by looking at the structure of the chromosomes surrounding the region that we think is involved in Tobiano. And as it turns out, there's a large rearrangement and inversion of a section of the chromosome that is probably affecting the regulation of some genes involved in production of pigment. So this chromosome inversion doesn't seem to cause many health deficits, but it is interesting from a biological and scientific point of view because it's a very unique situation. And polymorphic inversions and things like that are very hot in genetics right now, so it really elevates the horse to a new level as far as a model organism. Now, this inversion so far, from our own testing as well as that of collaborators that we have, it seems to be 100% associated with the Tobiano gene. So that means that it's going to be an excellent test, and we've designed a PCR, a DNA-based test that will enable us to look for this chromosome inversion and therefore predict which horses are going to have 2 copies of the chromosome inversion, 2 copies of Tobiano, known as homozygous, and are going to be true breeding. So these breeders can select for animals that are going to give them the best production of spotted horses.
And when and where can people see your paper on this?
This paper is going to come out in a scientific peer-reviewed journal called Cytogenetic and Genome Research. It is supposed to appear in the December-January issue. I think that's issue 119.
Great. Thank you very much for joining us today. Stay tuned to thehorse.com for more information on this story. That's it for this week's Ryder Report. I'm Erin Ryder, news editor of The Horse, your guide to equine healthcare. Visit thehorse.com for all the latest news on equine healthcare, management, and welfare. Have a safe and pleasant holiday, and we'll see you next week.